This week we will discuss about a small part, that is linkage recombination, sex determination plus pedigree chart. What is the use of pedigree and why we are using pedigree? Those things we are going to discuss this week. Last week some of the students have said that, sir, please take the class on board, but I am feeling some difficulties here because whenever I am focusing the camera, mobile camera, I have only mobile camera with me. So the clarity is not good. Whatever I am writing on the board is not clear or not visible when we are converted into some format. So please bear with me. And if you have any doubts, uh, please contact me. I will help you by giving the supporting the supported with the materials. So till the live classes begins, we starts, we have to go like this. So today or this week, for this week, we will be discussing about linkage and recombination. Till last week, we have discussed till the chromosomal theory of inheritance. Till the chromosomal theory of inheritance, everybody was sure that the chromosomes are responsible for the inheritance. So what is inheritance from that? That was 100 percentage did practically by Thomas Hunt Morgan. So he said that genes are transmitting from one generation to another. And one more thing he came to know from that. According to the Mendelian law of inheritance, one gene will control one character or one trait. That is what said. If there is one gene on one chromosome, it will control one trait or character. That was considered by the Mendel. So when the Thomas Hunt Morgan, what happened is when Thomas Hunt Morgan was actually he's an embryologist. He is working when he was working inside his lab. What he has found is he was using his experimental material, that is fruit fly, that is Drosophila melanogaster. He was using, the picture is down here, and we can see this Drosophila melanogaster in whenever we are going to the fruit markets. Whenever the fruit got ripened, you can see the small flies, small or big flies are coming there. That is clear there. So those are the flies he has took as the experiment. Not experiment, he just observed them. What he has observed is, apart from the parental phenotype with the white eye and the normal wings, he has found that some of the second generation there is some variation, there are some differences. So Morgan did his work, and Morgan said that one thing, what Mendel has said, that one gene controls one trait, is not possibly correct in all the cases. In some cases, genes are linked. Linked means both the genes are joined together or attached together. So, in that condition, genes can't assort independently. What is a third Mendelian theory, that is independent law of independent assortment. The characters can't independently assort, that means independently mix. So this is what this is what Morgan said. And for that he used this experiment, the whatever the experiment he has done over here, it is another version from the textbook that is given over here. That means uh, he has taken a parental character with the white eyes and the miniature wings, which is the female, and male they have taken the wild type, which is having white eyes and the wings, miniature wings he has taken. And when he crossed some of them, he got as in the recombinant. So he said that. But one of his colleague, that means who's worked with him, co-worker with him, His name is Stewart. Stewart was a scientist also, but working under Thomas Hunt Morgan. What he did is Stewart said that if we know the genes, where are they? For example, if some genes are present in chromosome number 1, if we know the character and if we know the position of the genes, we can determine that where these genes are located. So if we know where the genes are located, we can make a genome map. Actually, in the if you take one chromosome, where is the location of these genes, whether these genes are located together, or that means whether they are linked together or non-linked together. So for his colleague or his student Stuart, Stuart and Bridges, both of them did that. So by using this Mendelian experience, by using this Morgan experiment, scientists got a method to map the genome. That means to study the location exactly on the chromosome where it is. So this was one of the greatest breakthrough on those times, because there was no other methods to find out the location. We know about loci, we know about genome, everything, but exactly pinpoint where it is on the chromosome and the distance between two genes. They call it as centimorgan. And in humans, 1 centimorgan is equal to 1 into 10 to the power 6 base pairs. Approximately, it is considered. A base pairs means how many adenine and thymine, guanine and cytosines are linked together. That is considered as one base pair. One adenine, one thymine break bonding is called as one base pair. Like that, 1 into 10 to the power 6 base pairs are required, are considered as 1 centimorgan. So with that, the scientists started to map the chromosomes also, but it was not effective in all the cases, and it was not 100% correct also. And modern methods after 1960s was gone, introduced, and now we are running on this modern methods. And why is the reason why Morgan chose this Drosophila melanogaster? The reason is same what the Mendel did. That means easy availability, and it is very easy to grow this organism inside the laboratory. And whenever they are giving the offsprings, they will give many offsprings, so we can study it very properly. And size, that means they are very small, so we can grow this inside a bottle, or when we tie some bananas or something on the room, on the corner of a room, so they can grow very fast. So this is the main reason why they have chose this Drosophila as an experiment material. And next comes is sex determination. Sex determination, after studying about this gene and chromosomes and other thing, one of the main important thing is that how the organism is determining sex, whether it is a male or female. So there are mainly two three types of sex determinations are there. So if you're asking about the type of sex determination, these are the main part, that is XX and XY sex determination. So inside that XX and XY determination, we have Y-centered sex determination, X-centered sex determination, and XX, X0 sex determination, ZW, ZZ sex determination, then haplodiploidy. So these are the things. And next thing is the environmental factors. Environmental factors means as the chromosomes are determining the sex, the same way environmental factor also can determine the sex. But we are not going to study all of it. All of it we are not going to study, and we are much concentrated only on the basis of chromosomal sex determination as per our syllabus. So inside the syllabus, we will be studying about three types of sex determination. One is a sex determination in humans, sex determination in birds, and the sex determination in insects. So these will be the three areas we will be concentrated. Other areas we will not be discussing in the 12th standard, but actually it is there, it is present inside this world of other sex determinations are present. But for the examination and this syllabus sake, we will be discussing only these three. So we'll go one by one. So first we will discuss about this XY system of sex determination. This XY system of sex determination is very common in humans. One of the best example for this is humans. XY sex determination. That means the Punnett square has been drawn here. The Punnett square, according to this, we know that we have 46 chromosomes. That is 44 are autosomes, and XY is the sex chromosome. And when these sperms, that means the gametes, are formed, so there is a chance, 50-50 chance for a male or a female. And inside this X and Y determines the sex. That means XX is very easy to form, but Y gene has to become very closer, then only a male character will be determined. So Y is considered as in the XY system as a character defined. And this Y chromosome is, compared to X chromosome, it is very small. And this Y chromosome don't have many other characters X apart from this sex determination. There are no much things, very few are there, but X chromosome is very big, and the presence and absence, deletion of any pair from the X chromosome, there are a lot of diseases are also possible from that. And Y chromosomes also make some diseases, but it is rare. And when the Y chromosome diseases are possible, it will affect only to the males. It will never affect to the females, because XY makes the male and XX makes the female. So only the sons will be having the problem, and totally the females will be free. So that we'll discuss afterwards in the genetic disease. And next things comes is the haplodiploid sex determination. This haplodiploid sex determination, it is 50-50. That means in the haploid cases and the diploid cases, sex can be determined. So, and it is one of the most common factor that is found inside the honeybees. And how the sex is determined in honeybees, there are a lot of theories are running, many theories are running. And still now it is unclear, not exactly much clear, why these honeybees are showing, not only honeybees, many of the insects, why they are showing this type of difference. Because if you see this honeybee colony, the queen, that is having 32 chromosome, which is a female, this colony can determine whether they want workers now or drones. According to the need they can. How it is possible and what is the reason for that, it is not 100% clear, not fully understood. But we know this much, that the queen, which is a bigger in size, will be having the 32 chromosomes, and workers, which is having 30, workers also will be having 32 chromosome. But one thing is there, workers are sterile in nature, and the drones will be having 16 chromosome, which is male. So if you see here, this diagram, we can see that female and male, female is 2n in condition, which is diploid, and male is haploid in condition. And through the process of meiosis, females will produce two, possible to produce two gametes, one is N16 and N16. And male, through mitosis, they produce one. So one of this gametes, which is mating with the female, that is N16 and N16, will become diploid in condition, that is 2n is equal to 32. So here that will become a female, and this other one which has not received that male gamete will become n is equal to 16 as a male. And some conditions, the colony itself, if there are many females, they don't want many females, they need only one female at a time. Other females can be or it will be formed as workers. So workers are doing protection. They gathers honey, they protect the honeybee combs, then they will nurture the young bees, young laid eggs, and whatever the offsprings are coming out, they will be nurtured. And male also will support this. These are the main function that is taking place, and this is also one type of sex determination. And the another type of sex determination that we can see on birds, especially one of the best example we can take is as hen, cock and rooster and hen. So this type of sex determination, it is known as ZW sex determination. Actually, ZW sex determination, why they give a ZW sex determination? It is just to differentiate it from XY sex determination, XY type of sex determination, to make it a small difference. That's all. And if you see here the females are heterozygous in nature and males are homozygous in nature. You can see it is very clear. That means the male will be having two set of ZZ chromosome, and the female bird, that is a hen, rooster, will be having homozygous, and the hen will be heterozygous in nature. And when they are producing the gametes, so they can produce two males and two females at a time. So these are one of the sex determination pattern that is followed in most of the birds. So these are the main sex determination which you have to study according to your syllabus for the purpose of your examination, which you have to study. There are others also we are not going, because there are many fishes are there which can change the sex according to the pressure, water pressure, temperature of water, many things. There are a lot of things are there, so we are not entering into those things. Next we will move to the pedigree chart. Pedigree chart, this particular chart is used by the geneticians, those who are especially working in the field of genetics. And why they are using this chart is they use this chart to draw a family tree. Why they are using this family tree? The reason is to see whether which type of diseases or which type of characters are inheriting from generations to generations. For this thing, geneticians are using this. And this particular pedigree chart is not drawn for each and every person, but a person who is a biologist, who is a doctor, he or she can himself draw this pedigree chart. And this pedigree chart is used very much and helpful very much for seeing whether a person is carrying very different types of genes for some type of diseases and some type of characters. So they are used, and these are the symbols which they are used inside this pedigree chart. Actually, in this pedigree chart, they will not write, it is possible to write the name, but it will take a lot of space. So they have noted like this. That means if it is a circle without shading, it is called considered as a normal female. You can write the name there on top or down, you can write the name. And a square without shading is considered as a normal male. And if the male or female is affected with some type of disease, that will be 100 percentage will be filled, shading will be done. And if it is heterozygous, half portion of the male or the female will be filled. So if the half is filled and another half is free, not that is not filled, it is considered as that male or female are heterozygous in nature. Next come procipoitus. Procipoitus means it is a particular person, or we consider as a proband. This will be a particular person who was notified to genetician that he or she is carrying one type of gene can cause some disease. That means the first person who notified by the geneticians, or identified by the genetician, or brought towards the attention of a genetician because of some diseases, that is considered as the proband. And if there is any twins, so they will be labeled like this, fraternal twins. So sometimes there may be a chance that they may be having three girls, two boys. So instead of drawing many circles and squares, if the females are free, all the females are free, or all the females are equally affected, or all the females are heterozygotes in nature, so they can put the numbers inside circle, and inside if it is 3 it is considered as 3 normal girls, or square with inside the number it is considered as 2 normal boys. And if it is a diamond shaped, it is known as unspecified sex, their sex they don't know. And this is circle with a cut, known as deceased, that means dead, the person is no more. And if they have mated after before or after marriage or with anybody, so it will be represented by attaching a single line drawn between a male and a female. And if it is a consanguineous mating, that means the marriage is happened in between the same family, then it will be double line. And if it is an identical twins, they can use these type, that means two the twins can be joined by one single line. So these are the common symbols that is used for this pedigree. And I have given some questions also related to this pedigree. It is very important. Why? What is the reason is in the board examination, uh, they have just asked to draw a structure related to the pedigree, but in some cases they can ask how we are determining whether they are dominant or recessive, what are the cases for that. So if a pedigree chart is given, if a pedigree chart is given, see this, if a pedigree chart is given like this, from this pedigree chart we can determine some results, that means we can extract some results from it. The first result, that is that whether that particular especially this is used to find out the disease condition. So we know that we are having autosomes and sex chromosome, that is 44 number of autosomes and 2 of sex chromosome, that is X and Y, which makes 44, 46 X and Y or XX, which makes 46. So from which chromosome we are getting this particular disease, whether it is coming from a sex chromosome X or Y, or this is coming from an autosome, that means non-sex chromosomes, non-sex chromosome. And another thing, whether this particular disease will come when the genes are dominant or these genes are recessive. So these are the things we can find out from this. So there are certain set of rules are given here. For example, if we want to determine a person is an autosomal dominant, so there are some characters are there. So these characters are not easy to by heart each and everything, but when you put in this practice, so I will give a worksheet for this particular pedigree. So when we do that more and more, automatically without by hearting, this will stick to your mind, so it is very easy for that. So if it is an autosome dominant, so the conditions are both the parents should be affected. And when you come down, you can see that autosomal dominant, both the parents are affected. And the offspring, one of the offspring will be affected and the one of the offspring may be unaffected also. All the affected individuals must have at least one affected parents, that is true. If both the parents are unaffected, all the offsprings must be unaffected. So these are the conditions that is responsible for autosomal dominant. And if you're seeing for the autosomal recessive, if both the parents are unaffected and the offspring is affected, the trait must be recessive. For example, if you see here this autosomal recessive, one thing you can understand, if both the parents are not affected and one among the offspring got this disease, it means that the parents must be in heterozygous in condition. So heterozygous is the only condition where you will get one dominant trait, one dominant gene and one recessive gene. When these recessive genes got it, when the recessive genes comes, then only they will get the particular disease. That is the main thing you have to understand. And next one, if both the parents shows a trait, all the offsprings must also exhibit that trait. That's the same thing. And another one more thing, whenever these dominant things are coming, dominant means which is uh, expressed more. When these dominant characters coming, these dominant characters will never skip generations. That means it will be there in the parental generation also, it will be there in the first generation also, second generation also, third generation also. But it is not compulsory thing that everybody should have, some may be carrier also. But when it is coming to a recessive trait, it may skip generations. It may not be there in first generation, it will be there in the second generation. It may not be there in the parental generation, it may be there in the first generation. That you have to keep in mind. And next one, that is X-linked dominance. X-linked dominance means whether it is caused by a sex chromosome. So conditions for that. If a male shows a trait, so two must of all the daughters as well as his mother. So what they are saying that if the male is showing a trait, all the daughters and the mother must be affected or carriers. An unaffected mother cannot have an affected son. So unaffected mother itself cannot have an affected son. X-linked dominant traits tend to be more common in males. So where we don't have much evidences related to that. And another thing you can see that in this X-linked dominant, you can see that 100% incidence affected daughters from affected father. Affected father. If the father is affected, all the daughters are affected. You can see from that pedigree chart. And if it is X-linked recessive, if a female shows a trait, so two must of all the sons as well as the father. That means if the female is showing the trait, the parents males will be affected. An unaffected mother can have an affected son if she is a carrier. An X-linked recessive trait tends to be more common in males. Recessiveness will be very common in males because for the recessive trait to come, only one gene is required. And in males, one X chromosome is diseased, compulsory the male child will be having. So if they want to show in the females, then both the X chromosome should be there. So this is the main conditions I have given. I know that when you read this, study this, it will never come to your mind. I have given some pedigree chart to do by self. Do it, and if you have any doubts, please let me know. I will help you out how to do it. And next one is mutation. So mutation means sudden change inside the genome, that is called as the mutation. That can be a gene, that can be a set of codons, anything can be shifted or changed because of many things. So the substances which are causing these mutants are known as mutagens. And there are different types of mutations are there. Different types are there. One is known as the base substitution. You know that DNAs, next chapter only we will be discussing about DNA, then only we will be understanding about this much, what is this A, what is this D, etc. This DNA, whenever this mutation is causing in the base substitution, that means normal thing is that adenine, that is A, will pair only with T, and G will pair only with C. That is a normal rule. Sometimes instead of A is there, instead of T, it can pair with G. So these type of mutations are called as the base substitution mutations. And under this base substitution mutation, we may get some more examples known as the point mutations. For example, if you see here, AUG, it's a codon which codes for a particular amino acid which is responsible for making a particular protein. So this will be studying how the proteins are formed, etc. These things in the next chapter. So if and inside this point mutation, there are different types of point mutations are there. The first point mutation is known as the silent mutation. Silent mutation, you see here, AUG, GCC, TGC, AAA, CGC, TGG. This is the codon. But if you see inside the silent mutation, you see AUG, it is correct, no change. But you see the next GCT, but in top it was GCC. Balance all are same. But why it is called a silent mutation? Because it will never cause much change in the formation of proteins. That is why it is called as the silent mutation. And next you see it is known as a nonsense mutation, because no sense what it is doing. You see here AUG, GCC, TGA, and so on. A balance are there, and TGA, body don't know what to do with that particular sequence, whether to produce something else, they don't know. That is why it is called as nonsense mutation. And next comes is missense, that is AUG, GCC, then instead of TGC it is GCC. But what is happening? The amino acid that is formed here, instead of cysteine, it become arginine, which is called as the missense. It is missed the sense. That means which has to be produced, it is producing something else. That's the meaning. And next one comes is the frame shift. Entirely the three codons are shifted or deleted, or some added, division added or deleted or inversion, anything. And if you see here in the frame shift mutation, first you see AUG, GCC, it is the original codon, but when you comes here there is only G and C, it is gone. That means one C is gone. When that one C is gone, system will read as GCT, GCA, AAC, GCT, and when you comes to the last 2GG will not have a codon. So that is known as the frame shift. And next it's frame shift insertion. That means AUG is correct, GCC is correct, but instead of producing TGC directly, they have inserted one more C in between. So reading will go instead of TGC, that means the body will read not as TGC, the body will read along with C, CTG, CAA, like that. So it will produce another amino acid. That amino acid will code for another protein which may not be useful for us. And next one, insertion and deletion, both are taking place at a time, so that also will cause for a different formation of protein. If the proteins are formed like that, so this protein may not be useful maybe overexpression of some characters, or maybe it may leads to the formation of cancers also, maybe oncogenes, activation of oncogenes, etc. So the system itself, whenever the DNA is replicating and whenever the proteins are produced, the system itself have some methods to repair it. And what are the main reason for this type of mutation? One is DNA replication. DNA replication we are studying in the next chapter. So whenever one strand of new DNAs are formed, they should check it. That time normal case mistakes are possible. That mistakes has to be rectified. That is proofreading. If that error is not rectified, then it will cause to a mutation. Error in DNA recombination. That means when the match crossing over is taking place, that time chemical damages by taking some drugs, highly chemical drugs, and by going some radiations. In all these ways, this particular mutation is possible. So whatever it is given down, there are some type of mutations where it can takes place. And these are the questions which I have given for you. We have more questions also. So this particular questions you must do it and make it clear the pedigree. And if you have any doubt, please call me, put a message, I will contact you back. Thank you.